Let's start off with a simple concept. Before we had a protected environment, it was the natural environment that actually resulted in early mortality and morbidity and mortality, where there were hazards in the environment, predation, there was infections, we didn't have antibiotics, we didn't have vaccines. But today in our protected environment, what we're observing is that individuals are living longer, and what we're all interested in is not necessarily living longer, but living healthier for longer. And so the focus of this conference and many other conferences is to try to understand more fundamentals about the aging processes so that we might intervene in those aspects where we can. So let's start off with the definition. What is aging? Aging is associated with the gradual decline and performance and reserve capacity in organ systems. Now, organ systems are often associated with decreases in the number and function of cells within those organs. And as we age, old cells do not maintain and repair tissues as well as young cells. Everybody knows that, right? If you're a young kid and you cut yourself, your skin wound heals rapidly. If you're over the age of 60, it takes weeks sometimes for a superficial wound to heal as fast. Why is that? Because old cells do not maintain and repair these tissues as well as young cells. And this leads eventually to overall decreased vitality. So there's a lot of promising technologies here, and obviously in my overview I can't even touch the tip of the iceberg on all of these different technologies on this list. But many of these actually do, with the one near the bottom, the telomere extension cell rejuvenation, really does interfere, or I should say interconnect, with many of these other promising technologies, such as neuroendocrinology interventions, anti-inflammatory agents, antioxidant supplements, caloric restriction, the mitochondrial modulators, and stem cell therapies, all indirectly eventually come back to the ability of cells to proliferate and maintain tissues, and that's where the telomere technology is going to be important. So let's do a little History 101. So back in the 1800s, August Weismann stated that death takes place because a worn-out tissue cannot forever renew, and because a capacity for increase by means of cell division is not everlasting but finite. So that's how the field started, that they were explaining and separating the germline from the somatic cells. This was early in the history of cell biology, where we just discovered that tissues were made up of cells, and it was considered back then very common that cells were somewhat limited. They couldn't last forever. But then in 1921, another Nobel laureate, Alexis Carrel, who was at the Rockefeller Institute, made the following statement: all cells in culture are inherently immortal. The lack of continuous cell replication is due to ignorance on how best to cultivate the cells. Carrel and his co-workers claimed that they kept certain types of chicken embryo fibroblasts growing continuously for 34 years. And so that then became well established in the thought collective of science throughout the world, that if you take cells from the human body, they're immortal. They can basically grow forever. And that's where it was for quite a while, until the 1950s, when Leonard Hayflick, who was a cell culture supervisor at the Wistar Institute in Philadelphia, he was hired to run this facility, and because everybody had sort of believed Alex Carrel's notion that cells were immortal, his role was to go there and grow these cells and to keep them growing for research that was being done at this institute. And he couldn't keep his cells growing. He assumed it was his problem, that there was something with the cell culture constituents, the media, the glassware, because everybody had accepted the Nobel laureate's idea that cells were immortal. And then he made an interesting observation, that the cells always died at a particular time. It wasn't like sometimes they died after a few divisions, but they always went, these particular cell types, he could put them in the freezer, thaw them back out, and when he did that, they remembered how old they are. He called it the cell replicometer, in fact. So he had a problem here. He had to try to correct this. And so he said, what happens when I put cells in culture is there's a slow phase at the beginning called phase 1. Then the cells for a lot of days will grow robustly. And then there is a third phase where the cells actually slow down and eventually quit dividing. He made pictures of all this. And this is his classic published papers from his original published paper, wherein the cells in phase 1 look nice and youthful, and the ones over on the right where it's stated phase 3, they're large. And what's interesting is these large cells actually produce a different constellation of proteins compared to young quiescent cells. In both cases, these cells were not dividing. And so the question was, what's going on here? So he did a critical experiment. And even though this is hard to read, it was actually probably one of the more clever experiments that, as simple as it was, but it was very clever. So what he did is he took these fibroblasts that he was working with from both males and females, and he took some cells from a male that had divided 49 times in vitro. And he knew from his experience that they would only go another 10 doublings. So if there was something wrong with the media or something like that, then that would explain it. But then he added to the same culture female cells that were very young passage, and yet the culture didn't stop dividing at population doubling 60, and they kept going. But now there were no male cells left in the culture. They were all female cells. And so that eliminated almost all of the artifacts that one could have guessed were present why the cells would stop growing. It basically said there wasn't anything wrong with the media or the culture conditions, or there wasn't some rare virus or something in the thing that was causing the cells to die and stuff like that. So his only conclusion was that perhaps Alex Carroll's observations were incorrect. But then he had to overcome the thought collective. It's very hard once somebody makes a statement and says, this is the fact, how do you get the rest of the scientific community to believe you're right and a Nobel laureate is wrong? That's pretty tough, right? So he said that he thought what happened was that the way that Carroll actually kept his cells dividing indefinitely, because in those days they didn't have defined cell culture medias like we do today. Instead, they used to add extracts. They used to use chick embryo extracts and things like that. And it was Hayflick's hypothesis, the reason why those cells kept surviving is that he kept recontaminating the cells with fresh embryo fibroblasts from the chick embryos, the materials they were using. But anyway, that was his hypothesis, so he decided he had enough data to write his paper. He submitted it to the Journal of Experimental Medicine, and another Nobel laureate, Peyton Roos, said to him as they rejected his paper, the largest fact to have come from tissue culture in the last 50 years is that cells inherently capable of multiplying will do so indefinitely if supplied with the right milieu in vitro. And finally, as a good scientist, he didn't give up and just quit his career. He submitted it to maybe a less high-profile journal, but it was eventually published. And this was considered the beginning of the field of cellular gerontology. In fact, another Nobel laureate, Sir Macfarlane Burnet from Australia, coined the term, the Hayflick Limit, which is now well established in the scientific community. So to summarize what the Hayflick Limit really is, it says that normal human cells exhibit a limit to their ability to divide in cell culture and probably in vivo in people. Cells stop proliferating at a very predictable number of divisions, and that is dependent on the cell and tissue type. And upon attaining this limit, cells undergo a permanent growth arrest, which he coined the term cellular senescence. And this is really defined at the bottom here as an irreversible growth arrest. Sometimes people mix that up and say that when the cells get senescence, they die. But in fact, it's now been shown that cells that have reached that plateau, phase 3, where the cells are no longer dividing, can actually be kept viable if you give them the right nutrients for up to 3 or 4 years. So it shows that, in fact, senescence is an irreversible growth arrest, and they can be kept a long term if you give them the right constituents. So now we're going to switch to what is the mechanism. So the question that Hayflick was concerned with after that was, what is the replicometer? What is the timing mechanism within cells that made them always quit at a particular time? What was the counting mechanism? Nobody really understood that. So during that time, there was obviously other, again, I keep coming back to Nobel laureates. There were two other Nobel laureates that had been doing basic research. One was Herman Muller at the University of Texas at Austin, who was looking at X-ray mutagenesis experiments in Drosophila. The other was Barbara McClintock, who was at Cold Spring Harbor, working on cytogenetics in corn. Both of them made brilliant discoveries, and what they both came up with, and in fact Muller coined the term, telomeres, special functional complexes at the end of eukaryotic chromosomes. So what exactly are telomeres? Telomeres are these repetitive elements that are at the very tips of all of our chromosomes. We inherit from each of our parents 23 chromosomes. So you wind up with 46 chromosomes, and you wind up with 92 ends. Now, viruses and bacteria, many have circular genomes, so they don't need telomeres. But for some reason, most organisms have divided their genetic information up into little packages called genes, and they put those into a specific number of chromosomes, which vary from species to species. But there are ends of chromosomes. You've seen these pictures as they're illustrated on this slide many times. And if you can see the little green dots in the middle, those are the little tips on the ends of all these chromosomes. And what those tips are not genes. They don't code for things that make proteins or anything like that. Instead, they're just repetitive, monotonously repetitive, hexameric repeats of the sequence TTAGGG. And it happens over and over again. And in fact, about this time, another Nobel laureate, James Watson, who discovered the double helix, came up with the concept in 1972 called the end replication problem. And what that meant is every time a cell divided, you would, by the way cells, DNA replicates, you would lose a little bit of the ends of your telomeres every time a cell divided. In addition, it was believed that things like oxidative damage and other processing events could cause a gradual, progressive loss of these ends of our chromosomes. And it was believed when a few chromosomes or a single chromosome's telomeres got so short, that would signal an event that would stop the cells from growing. So if you think about it, our cells in our body are acutely prepared to deal with DNA damage. If you make a double or a single-stranded break, we have wonderful mechanisms to repair that damage. The problem is the ends of our chromosomes are protected, and they form a very special loop-like structure. And if they get too short, that loop-like structure may become unfolded, and when that happens, the cells see that as a DNA damage signal, and the cells stop growing. And that's what replicative senescence is about. So here's just a little cartoon showing the end replication problem. Every time a cell divides, you wind up losing a little bit of the ends of each chromosome. And that's actually a physical phenomenon that nobody argues with. And we can measure that. We can measure that in human beings. We can measure it in all sorts of organisms that have linear chromosomes. So an analogy that I like to use is like the plastic tips on the ends of a on a shoelace. These are called auglets, little plastic tips. So if you think about your chromosomes, all your 46 pairs of chromosomes and the 92 ends that we have in each of your cells, think about these little plastic tips as protecting those chromosomes from unraveling. And what happens when they get too short, they become unraveled, basically, uncapped, and that's what leads to the cells undergoing senescence. So what is the evidence that anything I've said up to this point with telomeres and senescence is real? Well, you have to go back and look at the correlations. And so over the last 15 years, there's been many correlations. We go back to the original Hayflick phenomena, which is illustrated here. Young cells divide a finite number of times, and then they stop dividing, as you see on the right. And Cal Harley and Carol Greider and other people in the early 1990s actually followed and measured the telomere length in cells between young and old. And the pictures you see at the bottom is called a TRF. It's a telomere restriction fragment, southern blot sort of mechanism that one can use to sort of track the shortening of telomeres with time. And because the telomeres in all of our cells are heterogeneous, Not all telomeres are the same, so when you actually look at telomeres, you could see, I think, pretty clearly on the left here in young cells that the spread of the telomeres is taller or higher than the ones on the far right that you can see over here in the old cells. So it was a correlation, but by no stretch of the imagination did that prove that telomeres caused the senescence. It was just a correlation. So what about in real people? There was another correlation from Robin Alshire's group and Nick Hasty, also in 1990. In high-profile journals published that if you look at the telomere length in fetal material or reproductive material, that on average when you're young or born, you have somewhere between 15 and 20 kilobase pairs. That's 20,000 base pairs of T2AG3 at the ends of all of our chromosomes. But if they looked at adult tissues, even adult tissues that rapidly divide, as you can see here from the GI tract or from the blood, as you increase your age, the length of your telomeres gets shorter and shorter. Again, this was a correlation that showed that telomere shortening correlated with increased age. Telomere shortening correlated with in vitro passage of cells in culture, but it certainly didn't prove that telomere shortening caused senescence. Okay? So now I'm going to switch, but I will come back to those correlations because it's very difficult in science to prove cause and effect relationships among almost anything. Most of what we do is test hypotheses, try to falsify them, try to collect data. But now, in order to make this story complete, what I'd like to do is to sort of switch a little bit and talk about what I call the double-edged sword, the relationship between aging and cancer. And I don't think I have to tell anybody in this audience that as we increase our age, the increased risk of cancer goes up. That most of us are protected except for the developmental pediatric cancers. Most of us are protected from getting cancer until we're in our mid-40s or later. And then we see this almost logarithmic increased risk of cancer until you reach about 85 where it sort of levels off. So what is going on here? And one idea, and it still is the hypothesis, that in fact replicative senescence may in fact be a very potent anti-cancer protection mechanism. If you think about it, in order to get cancer, you have to accumulate a lot of alterations within a single cell. While a lot of your cells may have alterations, most people don't get cancer. 2 out of 3 of us in this room will never get cancer, but 1 out of 3 of us will get cancer of some form. And the question is, why don't we all get cancer when we're 20 and 30 and 40? Why is it most of the cancers, I think the numbers are 70% of all cancers are in the 65 and older segment of the population. So that's actually a pretty dramatic percentage. And one way to think about it is that it takes divisions for some alteration in a cell to be retained. So if you look on the left here, you think about a single alteration, a single mutation, or a change in a cell occurs. Those cells divide up to 20 times, which is 1 million cells. And then within one of those 1 million cells, a second alteration occurs. And then a third alteration occurs. And if you look at the number of divisions that it takes, one can imagine that you might run up against the Hayflick limit, the senescence brick wall, as I illustrate on this cartoon, that might in fact initially protect us against the development of cancer. So is there any evidence for that? Well, the evidence is actually quite dramatic. Again, a lot of correlations. If you look at almost all pre-neoplastic, precancerous lesions like ductal carcinoma in situ for breast cancer, prostatic interepithelial neoplasias that's a precursor to prostate cancer, and the list goes on. When individuals have studied those pre-malignant diseases, they almost universally show that the pre-malignant lesions have very short telomeres. So what is that really saying? Are those short telomeres actually protecting us against getting advanced cancers, or are they actually starting the whole problem? An uncapped telomere might be driving genomic instability, leading to the initiation of cancer. So here's an image, if this will go forward. There it goes. So another way to look at telomeres besides this TRF approach that I mentioned earlier is called in situ fluorescence, in situ hybridization. And one can do this by having a labeled, a colored probe. Here, the telomeres are colored in green, And the centromere, which is the middle of the chromosome, is labeled in pink. And you can see on the top right what we call a metaphase spread. And in the bottom left is a cell that's an interphase cell. And one can actually go in there and look at how bright those green dots are relative to those pink dots, which don't change with time. So if telomeres are shortening, the green dots should get shorter and shorter, and whereas the centromeres don't get shorter with increased time or division, so you wouldn't see a change. So here's an example that Alan Meeker from Johns Hopkins provided me, where he just hybridized with a telomere probe, okay, no centromere, and they're all in pink in this particular picture. And what he shows here is this is a person with ductal carcinoma in situ. This is a major pre-malignant disease that increases a woman's risk for breast cancer enormously. And if you're detected with DCIS, they almost always surgically remove those, and you hope that you don't get a recurrence of the disease. What you see on this image is that the peripheral cells all have very bright pink dots, but these cells that are in the middle of this sort of duct-like structure have weaker telomere signals, suggesting that the luminal cells, where many ductal carcinomas arise, in fact have critically short telomeres. And in fact, if you look at recent publications and they measure how short your telomeres are, you look at the shortest quartile, the second quartile, the third, or the fourth, which would be the longest, and you look at women that are at increased risk for the development of cancer. Here you can see BRCA1, BRCA2 are major predisposing mutations that lead to an increased risk of breast cancer. But in all of these cases, what you see in the black bars is that those women with the shortest quartile of telomeres actually had the highest increased cases of cancer. Okay, so it really says while the short telomeres might have been protecting them initially, eventually those short telomeres lead to an increased risk of developing cancer as well. And another study that's been published just recently in 2010 looked at a variety of cancers where they showed, again, that the cancer incidence was higher in those individuals with the shortest telomeres, and the overall survival, overall mortality was also worse in those with the shortest telomeres. Again, correlations on top of correlations showing short telomeres lead to senescence and an increased risk of the development of cancer. So this is what this all came down to. This came down to this, what we call the telomere hypothesis of cancer. Most premalignant tissues have short telomeres, and therefore while telomere attrition may initially be a tumor suppressor pathway, that in combination with other cellular alterations, those short telomeres may actually drive the genomic instability that we know is a hallmark of cancer. Now the problem is, how do the cancer cells overcome the telomere problem? We've been sort of waltzing around this, that somehow cancer cells are almost always able to continue dividing and spread throughout the body, which is really what is the major problem with cancer. So how do cancer cells avoid this problem? They have to engage some mechanism to stabilize those telomeres. And we and other groups now for the last decade have been studying this greatly. Okay? And in fact, brilliant scientists like Doug Hanahan and Bob Weinberg's group published last year what they called the hallmarks of cancer. And while most of you probably can't read that from the back, down at the bottom in blue there it says one of the hallmarks of cancer is enabling replicative immortality, saying that somehow cancer cells have to be able to overcome the Hayflick limit. How do they do that? That's the question. And it turns out they do that by activating a very interesting enzyme. It is the only cellular reverse transcriptase that we as humans have. There are obviously viral reverse transcriptases, but this is the only reverse transcriptase, and it's called telomerase, or terminal telomere transferase. And it turns out that telomerase is detected in almost 90% of all human cancers. So this turns out to be the real mechanism on how cancer cells bypass the Hayflick limit. And it does this because telomerase, as is illustrated in this little cartoon that's moving on the screen, that telomerase is basically a molecular motor that adds new telomeric repeats onto the ends of chromosomes. This is the way that cancer cells bypass this, by upregulating or turning on a quiescent telomerase gene that was clearly important during early human development. And in fact, we know that if you look at reproductive tissues and early embryonic tissues, they are full of telomerase activity. And then as we age, tissue by tissue, the telomerase gets turned off, and it stays off unless you get cancer. And we know a lot about the components. We've got new assays that are very sensitive called the TRAP assay. We can do all kinds of molecular gene jockeying to measure and quantitate all these things. But the question is, telomerase is detected in 90% of cancers. In fact, it was such a well-excited field that the originators, the people who first discovered this, Elizabeth Blackburn on the left and her graduate student Carol Greider on the right, and I'll have more to say about Carol Greider in a few minutes, received the Lasker Award in 2006, and in 2009, again, another Nobel Prize for work that's been done in the telomere field. And their prize was for the prediction and discovery of telomerase, which they did in a small single-cell protozoan, and they've worked on things like yeast and things like that to study this in detail. And they discovered this telomerase, a remarkable RNA-containing enzyme that synthesizes the telomeric repeats on the ends of chromosomes, protecting them and maintaining the integrity of the genome. So now we've got the world's greatest leaders and people agreeing that telomeres are important. We know that telomeres get short and you get old. We know that if you get cancer, there's this enzyme that can turn back on that can keep your cells growing. So a central experiment that our lab was involved in doing, also about a decade ago, was to find out if, in fact, telomerase was, in fact, causal in the Hayflick limit. And what better way to go from a correlation to cause and effect than by saying if cells have short telomeres and you put telomerase into those cells, do they in fact still go through do they stop at the Hayflick limit? And the answer is they don't. All we had to do is put this one gene, hTERT, the telomerase reverse transcriptase, into normal Hayflick-like cells and put those in. We turn on telomerase activity. Telomeres now start elongating, as I think you can see as the little average goes up here in the middle chart. And in fact, instead of cells undergoing the Hayflick limit, they continue to divide. So now the question you should be asking yourself is, are those cancer cells? Is putting telomerase in going to cause these cells to become cancer cells? And the answer is absolutely no. Okay? So we've done that over and over again. These are the hallmarks of cancer. You know, cancer cells lose contact inhibition. They require fewer growth factors for growth. They can grow what we call anchorage independent. They don't have to grow on a substrate. They have lost cell cycle checkpoints that tell them when to grow and not to grow. But if you look at hTERT immortalized, telomerase immortalized normal cells, they look just like normal stem cells. Okay? The only difference between normal somatic stem cells, adult stem cells in the body, and hTERT immortalized somatic cells is that the hTERT cells divide indefinitely. They don't shorten their telomeres. Only one other type of cell that we ever have that grows indefinitely as stem cell is called an embryonic stem cell. So human embryonic stem cells actually have robust telomerase, and they fully maintain their telomeres. And we know embryonic stem cells can make a perfectly normal human being. So we know that it's not a requirement of being a cancer sort of phenotype. So we're left with this dichotomy, you know, come on, it's either one or the others. So here I'm talking about aging. We're either going to get old and have all the problems associated with aging, or we're going to get cancer. Is there something in between? Can we slow down the aging process? Can we prevent the cancer process? Well, of course. Obviously, if telomerase is on in so many cancers, we should be able to find something to inhibit telomerase, right? And we've done that. So I call this the two faces of telomerase, the Dr. Jekyll and Mr. Hyde of telomerase. On one part, there's the telomerase is turned on in 90+% of cancers, and that is a cancer-permissive situation. And then one can say, well, what about putting telomerase or some small molecule that can activate telomerase into normal cells? Would that be important in some sort of aging cell engineering, cell rejuvenation that might in fact keep our cells going longer and better without an increased risk of cancer? Now, an interesting experiment that we did was with a certain type of pediatric cancer where we observed that certain children were born with this disease called neuroblastoma. This is a cancer type that's on the top of the kidneys and the adrenal gland. And when these children are born, it turns out, and you might be asking, why is it only 90% of the people get telomerase turned on in cancer? Well, here was a very interesting observation that we made, and that is these particular children, and we wish this was true in adult cancers, but in certain pediatric cancers, there is these interesting correlations of tumors regressing and people going into spontaneous remissions of their cancers. And neuroblastoma is one of the sort of showcases of this, where these young children are born with a third of their newborn body weight being metastatic cancer. And they don't know what to do with these kids because they don't want to give them chemotherapy, they don't want to give them radiation therapy because they're so young they'll wind up with secondary cancers when they're teenagers. Instead, they just do whatever surgical procedures to keep their organs functioning. And as you can see in the top in this Kaplan-Meier survival curve, that a huge number of these kids go into spontaneous remissions of their cancers, and a few don't. And it turns out that correlation is directly on whether their cancer cell turned on telomerase or not. If they didn't turn on telomerase, those cells eventually hit the Hayflick limit, and they quit dividing. And then they were recognized as being foreign cells out in the interstitial spaces, the liver, wherever the cells had gone, and phagocytic cells in our body cleared those cells. The kids that had telomerase activity, their cells continued to divide, there was no inhibition, and they have a very poor overall survival. But if there's one point about this, there's 2 points about this slide. One is it shows you don't need to have telomerase to make a cancer, right? So that could explain a big chunk of those 10% of cancers that don't have detectable telomerase. But more importantly, it suggests in order for a cell to continue to divide to eventually lead to an advanced malignancy that leads to the death of a patient, you need to have a mechanism to overcome the Hayflick limit, and that's usually by turning on telomerase. And now if you're interested in therapies, you would say, therefore, any way we can inhibit telomerase in these children with advanced cancers, if we could come up with specific anti-telomerase therapies, would we be able to have a major impact on these 90% of people with telomerase-positive cancers? And the answer is we're making great progress. I can't go through all the details, but there's now phase 3 and a phase 2 trial, different approaches. There's immunotherapies against telomerase. There's small molecule inhibitors. There's a lot of excitement in the field, and this has been going on for 15 years almost. I mean, telomerase was discovered in 1997. Well, it wasn't discovered. It was cloned in 1997 by Bill Andrews, one of the people here sitting on the front row when he was with the Geron Corporation. And we now have these robust inhibitors that are in advanced clinical trials, and the hope is that these will be used in a way in an adjunct way with standard therapy for cancer patients, hoping that if we could give you these telomerase inhibitors, you will drive the telomeres short, and we now know from all the preclinical experiments that the cells will eventually undergo cell senescence and apoptosis, because once they become a cancer cell, they no longer respect the Hayflick limit as much, and when you drive those telomeres short, the cells almost all undergo apoptotic cell death. So it turns out that there is now increased evidence in a whole variety of human diseases where telomere biology is becoming important. We know people with diabetic ulcers, for example, chronic wounds that don't heal, have critically short telomeres. Is that explaining why these wounds don't heal as well? There's myeloproliferative disorders where you have a decreased immunity. Macular degeneration is another thing. It's the leading cause of age-associated blindness. The list goes on. The vascular endothelium. You always hear about plaque building up, but what it does is it causes the vascular endothelium to turn over more in your coronary arteries and things like that. And maybe when cells get old, they're more conducive for plaque attaching to these sort of things. And then there's the premalignant diseases. And then there's diseases like dyskeratosis congenita, which actually have mutations in some of the components associated with telomerase. And there's another disease called idiopathic pulmonary fibrosis, which is associated with mutations in the telomerase genes. What does that mean, having a mutation in the telomerase gene? So here's sort of the cartoon of the components of telomerase. It is a ribonucleoprotein holoenzyme. It has the TERT, or the telomerase reverse transcriptase component. It has this black squiggly-looking line called HTR, which is the telomerase functional RNA. And then you've got associated proteins associated with it. And we've now discovered that the whole new field of pathology called the telomeropathies, where certain people with things like pulmonary fibrosis, bone marrow failure, things like dyskeratosis congenita are due, in fact, and occur in younger people because they are born with mutations in telomerase, which means that they don't have the full complement that most of us in this room have. In other words, we're born with 15 to 20 kb of telomeres. These individuals are born with about half that length, so therefore they're getting short telomeres much earlier in life. Here's an example of a study that we did with Christine Garcia at UT Southwestern, where we actually just looked at the peripheral blood mononuclear cells from patients from families with where this pulmonary fibrosis ran through the families. We called it idiopathic because we didn't know what the cause was. Now we do. And as you can see, there's this one person there in the red square who has a mutation in a telomerase gene. And look at her. She's 52 years old, really critically short telomeres. But if you look across at the blue square over here on this family pedigree, you'll see here's a 26-year-old that actually got the mutation, but she doesn't have the disease yet. 26 years old, we can tell her that in 20 years she's going to probably get idiopathic pulmonary fibrosis, and there's nothing we can do for her. We could tell her to not smoke. We could tell her to take some antioxidants maybe, but we're not even sure that that will do any good. And yet we can predict this based on her telomere length. So what this really says is that mutations in components of telomerase may lead to premature stem cell depletion. And in these diseases, what happens is eventually that leads to clinical disease. And what it says most importantly is that haploinsufficiency in telomerase leads to premature stem cell depletion and clinical disease. And more importantly, it says we do not have an excess of telomerase. That for some reason, being a long-lived species, we regulate this gene extremely carefully, and we don't want to turn it on at too high a levels because of the increased risk of cancer. On the other hand, we need enough telomeres to last us a full lifespan. And so we're sitting here on this double-edged sword between aging and cancer, as we've already mentioned. So another way to sort of look at this in cartoon sort of form is that normal tissues and organs gradually shorten their telomeres throughout lifespan, and eventually, as you reach this pink area, you run into some sort of increased risk of disease. Those individuals with the telomeropathies start off with shorter telomeres, and therefore they reach this pink area where the disease risk increases at a much earlier age. So that should be telling you something about maybe there's something we can do for these individuals if we could just re-elongate their telomeres.